All right, ladies and gentlemen, welcome back. We are on to our final topic in genetics, and that's to discuss these charts called pedigrees. So, a pedigree is much like a family tree, but we're using them in terms of genetic characteristics and using them to study the way those traits get passed from one generation to the next. So, these are particularly useful to scientists or genetic counselors who are trying to understand the way a trait gets passed on from parent to offspring. And they're great tools, but they've got some serious limitations. Um, First off, they're great to use when we talk about characteristics in organisms where doing some sort of test cross just really isn't a viable option. So remember, those test crosses are taking an individual who is showing a dominant trait, but we want to know whether or not that individual is homozygous or heterozygous. So if I were capable of doing a test cross, I would take that individual who is dominant for the characteristic, mate him with somebody who is recessive, because then I know that person is homozygous, and I would study their offspring. If at any point any recessive individuals popped up in their offspring, then I knew that that person of questionable dominant genotype had to be heterozygous, because that's the only way you can get a homozygous recessive individual from that particular cross. If they never produce a recessive offspring, then I know they should be homozygous dominant. That being said, I can't necessarily go and do a test cross with a human. The ethical implications of that are huge, right? I can't just come up and tell you, hey, I want you to mate with that person and let's see what happens to your offspring. That's just not going to go over well. Two, you need to be able to produce enough offspring in a short enough period of time to actually study this. Remember that Punnett squares are only predictions. I need enough offspring to actually get a ratio that would be close to what we would predict in that Punnett square. I can't do it from two or three individual offspring. I need a large pool to sample from. Once again, not really something viable from a single human cross. So test crosses in humans just really not going to be an option. That being said, pedigrees really are only useful if I've got large families, because I still need a lot of data, and two, only if that family has kept really good records of characteristics over long periods of time. So I need to be able to produce a pedigree from somebody who knows a lot about their family's medical genetic history. And more often than not, that's getting harder and harder to do. So while pedigrees are still an option, with genetic testing and DNA screening getting better, cheaper, and more reliable, these pedigrees are starting to become less of a mainstay tool in your human genetics, at least. But they're still there, so let's take a look at them. So here's some examples. All right, this is a pedigree that was produced. So if you look, there's very clear and consistent symbols being used, connections between families. And if you've ever seen a family tree, hopefully this looks relatively similar. People's names are there. Their relationships to each other are also well established. And you very often end up seeing a key connected to these pedigrees. Do yourself a favor anytime you create one, create a key. Anytime you're reading a pedigree, make sure you take some time to look at those keys, because although I'm going to share with you today some conventional symbols, that being said, each symbol isn't always used the same way by every person who creates that pedigree, so you want to make sure that you understand what their symbols mean in that particular set of information. Here's another one. This one's of the royal family. So there's Queen Victoria. All right, and if we take a look, the more and more we come down here, we see all of their children and their connections. And if you know anything about the royal family, they have a horrible reputation, if you will, of keeping those bloodlines quote unquote clean. But really, we're just talking about a bunch of incest. And so if you start looking in here we get all sorts of hemophilia showing up in their male offspring as a result of all that incest. The more you mate with the same family, the more likely you get those less desirable recessive traits to show up more frequently. So here are some pieces of information to understand when you're reading and constructing pedigrees. Again, my first goal, or my first tip, I should say to you, is always to make sure you create the key and also read the key if you're reading someone else's pedigree. But some conventional symbols. These are pretty consistent. Circle is always a female, square is always male. These are lines that may or may not get used. So if I'm talking about a set of parents, so here's dad and mom here, all right, and they're connected by a line. If I look here, you may see fraternal twins get separated off like this so that we know that they're born at the same time, but they're fraternal. So genetically speaking, they're not any more similar to each other than another set of siblings that were born at different times. But identical twins are ones you really do want to connect. And so you may see a set of identical twins set up like this because they are genetically identical. They've come from the same egg, same sperm, so same fertilization. Just as that egg was forming that embryo, it happened to split completely and formed two embryos as a result. So genetically, those identical twins are really ones you want to keep track of because they are, in fact, genetically identical. They're clones of each other. Those fraternal twins are not. So while you may choose to designate that they are twins, they are not going to be genetically identical because it's two eggs, two sperm, unlike the identical set of twins. Couple other lines you're going to see, and these are pretty standard. Horizontal line between a male and a female is what they call a marriage line. Probably a little old school term here, but really all we're talking about is a married couple, or even more, just a set of parents that have children. Your siblings then will come down from that, and you'll get these horizontal sibling lines. So these are all of this couple's children, and you can see that they are all connected as a result of that. Some other terms, and here's where you may or may not see these consistently used. I would say this one's pretty common. Your affected individuals are colored in. Your carriers also probably more often than not shown this way, where you show half of them shaded to indicate that they do carry a trait, but they're not actually showing it. You may see this as an X-linked carrier symbol. Again, pay attention to the key, see if they're using it or not. And this one may also show up if they choose to designate that an individual in that tree is in fact deceased. They'll put a slash through their symbol. So here's the big thing. How do you determine when you're looking at a pedigree whether or not the chart that you've created demonstrates a certain particular inheritance pattern? Well, let's start off here. So kind of break it into two questions. First and foremost, is the pedigree that you're looking at autosomal, or is it X-linked or sex-linked? So when I look at pedigree charts, the easiest way to understand this is to look at the difference between male and female. If I'm overwhelmingly seeing that males are the ones affected by the disorder, then it's more than likely that it's an X-linked or a sex-linked disorder. That being said, keep in mind that it doesn't mean that a sex-linked trait can't affect some females. It's just that you will disproportionately see a larger amount of males affected than you will females. Go back to this one right here, right? So if I take a look, overwhelmingly we see males are the ones affected. All right, not that females could never have been affected by it, but you just don't see it very commonly. The next question then is, if it's not sex-linked, then it's got to be autosomal, in which case you'll see a much closer 50-50 ratio between men and women who are affected by the trait. So if I take a look at this one, is it autosomal or is it X-linked? Well, hopefully you're able to come up with the fact that it's autosomal. If I look, I've got 6 total individuals in this particular pedigree that are affected, 3 who are male, 3 who are female. So this one's really obviously cut and dry autosomal. Once I've determined whether it's autosomal versus sex-linked, then I want to know, is the disorder or the trait that I'm tracing dominant or recessive? If it's dominant, then at least one of the parents must have the disorder or the trait in order to continue seeing it. And so oftentimes you'll see this show up in virtually every generation. If it's recessive, on the other hand, neither parent has to have the disorder. They can both be heterozygote carriers for it and still end up with a child. So this is a situation where you might have it skip generations. So if I take a look at this one, am I expecting this to be dominant or recessive? And more than likely, this is dominant. I see in each set of parents, one parent is affected as well as some of their children. So I see it in every generation. So it's probably a dominant trait. How about this one? This one has to be recessive. If I look at that first generation, nobody's affected. So they're passing it on to their children, who then in turn also do pass it on to their children. So it does show up in those bottom two generations. But because I have that first generation with no parents affected, this has to be a recessive characteristic, which means all sets of grandparents, in this case, all parents in that first generation have to be heterozygote carriers for this particular disorder.